Hello, my name is Bill,
And my name is Tommy.
and today we are going to talk about a very interesting thing called dopamine.
In 1910, George Barger and James Hill were the first to discover the 3,4-dihydroxyphenylethylamine at the Wellcome Laboratories in London. Then an English pharmacologist named Henry Dale suggested the shorter name of dopamine in 1952. Finally, a Swedish neurophysiologist named Arvid Carlsson found dopamine to play an important role as a neurotransmitter.
This neurotransmitter is synthesized with enzymes called tyrosine hydroxylase and dopa decarboxylase. It is an amino acid derivative that is made from tyrosine and belongs to the catecholamine family. Within the catecholaminergic neurons, tyrosine hydroxylase adds a hydroxyl group to the para position of phenylalanine, then meta position of tyrosine, yielding L-dopa. L-dopa is then rapidly converted into dopamine by dopa decarboxylase. There is no genes that code for dopamine because it is derived from an amino acid. And because dopamine does not have genes to transcribe, the post-transcriptional or post-translational modifications cannot occur.
Dopamine activation can be triggered by electrical impulses that come from action potentials. When the electrical impulse reaches the axon terminal, it causes the release of the neurotransmitter dopamine into the synapse. This in turn causes the dopamine to travel to the receptors of the postsynaptic neuron.
Dopamine is widely circulated in the central nervous system and some peripheral areas such as the cardiovascular and renal system. In the brain, some of dopamine's functions is involved in the control of cognition, movements, emotions, memories, reward mechanisms, and the regulation of prolactin secretion by the pituitary gland.
When there are low or high amounts of dopamine, it triggers a negative feedback loop, which in turn maintains homeostasis. One method in which homeostasis can be shown is through the nigrostriatal pathway. This pathway is one of the major dopamine pathways that helps in movement. When there is a loss or decrease in dopamine in this pathway, more dopamine is made, triggered by the feedback loop. If no dopamine is released, the loss of dopamine can cause Parkinson's disease, which also affects movement.
Now that we've talked about some of dopamine's functions, we can talk about where it is produced.
Dopamine is produced in the dopaminergic neurons in the ventral tegmental area of the substantia nigra, the midbrain, and the arcuate nucleus of the hypothalamus.
In the periphery, dopamine is found in the kidney where it functions to produce renal vasodilation, diuresis, and natriuresis. One main function of dopamine occurs in the nigrostriatal pathway. Dopamine in that pathway is essential for voluntary motor activity. Dopamine itself works by acting on two primary receptors, the D1-like and the D2-like receptors.
Other parts of the brain in which dopamine acts on includes the hypothalamus, the olfactory bulb, the substantia nigra, the ventral tegmental area, and in the periaqueductal gray.
As previously mentioned, before dopamine can act on a specific cell, it must first bind onto dopamine receptors. Those receptors include D1-like receptors as well as D2-like receptors. D1-like receptors include the D1 and D5 receptors.
Binding to the D1 receptors allow voluntary movements, regulates growth and development, feeding, affect, attentions, working memory, learning, reward, sleep, impulse control, reproductive behaviors, and control of renin in the kidney. D5 receptor binding is involved with the pain process and endocrine functions related to dopamine.
D2-like receptors include D2 through 4 receptors. D2 receptors permit regulation of blood pressure, vasodilation, and locomotion regulation. It's also involved in working memory, reward motivation, renal functions, and gastrointestinal motility.
D3 receptor binding causes increased endocrine function, regulations of locomotor activities, as well as one's emotions. D4 binding allows for regulation of renal functions, gastrointestinal motility, vasodilation, blood pressure, and various cognitive functions.
As for D1 receptor locations, it can be found in high concentrations in the mesolimbic projection, olfactory bulb, striatum, substantia nigra, nucleus accumbens, caudate, and the putamen. It can also be found in low amounts in the hypothalamus, hippocampus, thalamus, kidney, and cerebellum.
The D5 receptors are found in the substantia nigra, sympathetic ganglia, hypothalamus, hippocampus, kidney, heart, blood vessels, adrenal glands, and the gastrointestinal tract. As for the D2-like receptors, the D2 receptor are found mainly in the ventral tegmental area, olfactory bulb, caudate, substantia nigra, nucleus accumbens, and the putamen. They could also be found in the cortex, hypothalamus, sympathetic ganglia, septum, heart, kidney, blood vessels, adrenal glands, and the gastrointestinal tract.
D3 receptors is only found in the central nervous system and found specifically in the nucleus accumbens and the olfactory bulb. D4 receptors are found in the substantia nigra, amygdala, hippocampus, thalamus, hypothalamus, frontal cortex, sympathetic ganglia, kidney, heart, blood vessels, adrenal glands, gastrointestinal tract, and the globus pallidum.
The signaling pathways for dopamine can be divided into four major pathways in the ventral tegmental area of the midbrain. One of them is called the mesolimbic pathway, highlighted in blue. It carries dopamine from the ventral tegmental area to the nucleus of the amygdala and hippocampus.
The mesocortical pathway, shown in red, connects the ventral tegmental area to the frontal lobe of the prefrontal cortex. It is important for normal cognitive function of the dorsolateral prefrontal cortex and may also be involved in motivation and emotional response.
The nigrostriatal pathway in green spreads from the substantia nigra to the neostriatum, which is involved in the production of movement as part of the system called the basal ganglia motor loop.
Finally, the tuberoinfundibular pathway runs from the hypothalamus to the pituitary gland where dopamine is released and regulates the secretion of prolactin.
When L-dopa leads to increased dopamine levels, this overproduction can lead to toxic effects and the production of free radicals. Free radicals are generally made under the normal human pathological conditions.
Free radicals have an unpaired electron, making it highly unstable and reactive. This instability causes the free radicals to interact with other cellular elements. The highly reactive hydroxyl radicals, especially when made excessively, can cause cellular damage and may lead up to cell death.
In normal cases, when there is excessive dopamine, the release of GABA neurotransmitters inhibits the release of dopamine by binding to GABA receptors. Certain drugs can lead to an overproduction of dopamine, such as opiates. Opiates bind to opiate receptors, and with the opiate receptor activation, it causes GABA release to be decreased. GABA is what inhibits dopamine release, and with lower amounts of GABA, more dopamine will continue to be released. Naloxone can be used as a cure as it acts as an opiate receptor antagonist. It binds itself to the opiate receptor as it has a higher affinity, allowing it to bind. By doing so, it blocks further opiate binding, reversing its effects.
An underproduction of dopamine can result in hypoactivity, which is the decrease in behavioral or locomotor activity such as decreased food consumption.
Potential consequences of a lack of dopamine can lead to diseases such as Parkinson's disease. Parkinson's disease is a neurodegenerative disease that affects dopaminergic neurons concentrated in the substantia nigra.
There are no cures for Parkinson's disease, but there are treatments and medications for the symptoms, which includes tremors, bradykinesia, uncoordinated movements, and limb rigidity. This disease affects mostly the elderly, as it takes a long time, usually over years, for the symptoms to develop.
There is a synthetic dopamine, but it cannot be used for treatment because when consumed, it cannot pass through the blood-brain barrier. Instead, a dopamine precursor is used called levodopa, and this can be used for treatment instead. L-dopa, or levodopa, is capable of being converted into dopamine in the human body, which will eventually help treat movement problems such as Parkinson's.